When a virus replicates, mutation in its genetic sequence occurs, which may alter some characteristics of the virus. If the altered virus mutated significantly enough from the original virus and gained survival advantages through these mutations, it may spread rapidly in a host population, becoming a new variant. The SARS-CoV-2 Delta variant is a mutated strain from the original SARS-CoV-2. It includes these strains. The Delta variant was first found in October 2020 in India. As of July 2021, it has spread to at least 92 countries with a cumulative prevalence worldwide of 7%. While in India, it has a higher cumulative prevalence at 35%. On 11 May 2021, the Delta variant was reclassified from a variant of interest to a variant of concern. To know more about the differences between these classifications, feel free to watch the video for SARS-CoV-2 variants on my channel. What are the key mutations acquired by the Delta variant that makes it a variant of concern? From the genetic sequencing of many virus samples from infected individuals, it was discovered that the Delta variant has accumulated over 21 mutations with about 7 mutations on the spike protein. This information is of great significance as the spike protein is involved in both the transmissibility of the virus and the immune response by the host against the virus. This is the general structure of the SARS-CoV-2, and the red protrusions on the surface of the virus are the spike proteins. For the SARS-CoV-2 to replicate in host cells, it must first use its receptor binding domain on the spike protein for attachment to the cells. In addition, the cell surface protease TMPRSS2 facilitates the cutting of a part on the spike protein, called the furin cleavage site, allowing the virus to enter host cells for viral replication. Moreover, most antibodies generated through infection or vaccination are stimulated by the spike protein. In other words, the immune system of the host eliminates the SARS-CoV-2 by recognizing the spike protein. From the list of mutations on the spike protein, a few mutations have been extensively studied to understand their impact. Similar to the other variants of concern including the alpha, beta and gamma variant, Delta variant has the D614G mutation which increases the transmission rate of the virus in the community. To know more about the impact of D614G, feel free to watch the video for alpha variant on my channel. Moreover, the L452R mutation further contributes to the increase in transmission rate of the Delta variant. This is due to the structural change on the spike protein caused by the substitution of the amino acid leucine at the 452 position in the chain to the amino acid arginine, causing stronger interaction between the spike protein with the ACE2 receptor on host cells. It was observed that the combination of L452R and D614G mutations increases viral entry into host cells, leading to higher viral load and transmission rate, which is the characteristic of the Delta variant. In addition to that, structural change of spike protein by the L452R mutation also leads to immune evasion of the virus, as shown by the decrease in effectiveness of antibodies in targeting the virus. Other notable mutations of the Delta variant include the T478K and P681R mutations with amino acid substitutions as listed. T478K mutation has been linked to increased viral transmission rate while P681R has been shown to increase cell fusion, which might affect viral infectivity. Both mutations also contribute to viral immune evasion. In general, the mutated characteristics of the Delta variant include more easily spread among the community, including household transmissions, much more than the Alpha variant, which led to its reclassification. Some suggest that the rate of airborne transmission is heightened with infection occurring within 5 to 10 seconds, without physical contact, compared to the 15 minutes for other variants. Delta variant is also more transmissible across all age groups, and is considered the most transmissible variant so far. In addition, increased resistance towards some emergency use authorization antibody treatments have been observed. Examples of vaccines that conducted studies against the Delta variant include Pfizer, AstraZeneca, Moderna and Sinovac. Current available vaccines are still effective in preventing the serious complications of COVID-19 including hospitalizations and deaths with some reduction in efficacies. Vaccines not listed by no means signifies non-efficacy, but rather due to limited available data. Vaccines listed also by no means signifies lesser efficacy than non-listed vaccines, but rather due to the available data from studies conducted. Therefore, this provides evidence that although current vaccines have reduced efficacies, 2 doses of these vaccines are still effective against the Delta variant with more than 50% efficacy, which is the efficacy cutoff point set by the Food and Drug Administration. If you enjoyed this video, don't forget to like, share or subscribe to my channel. If you would like to learn more about other variants of concern, check out this series.